Hello and welcome. Today we are going to discuss about Hardy-Weinberg law. Okay, it is also called as Hardy-Weinberg Equilibrium. And it is a very important topic in population genetics. So what does this Hardy-Weinberg law states? It simply states that relative frequency of alleles remain constant, which means alleles which are different forms of genes in a population remains constant generation to generation in a population. Okay, where organisms are sexually reproducing, which means in a population which is reproducing sexually, the alleles which are transferred from parents to offspring remain constant generation to generation. There is no loss of alleles, there is no change in this frequency. When only it is applicable when the population is big, organisms show random mating. When the population is big, Hardy-Weinberg equilibrium law is applicable. When the organisms show random mating and there is no actually rising of no any new mutation, any sudden change in the genetic makeup, and organisms survive and have uniform reproductive potential. So Hardy-Weinberg equilibrium is only applicable when the population is big, organisms so random mating, individuals are random mating. There is nothing like goal oriented. There is nothing like directional, random mating, and no any new mutation. And organisms reproduce and they survive have uniform reproductive potentials. Okay, so these are things when only this Hardy-Weinberg law is applicable. So we have a equation also here. Here the particular dominant allele is represented as P, and when it is in pair, it is represented as P square. In the same way, the recessive allele is represented in Q, and when in a pair, it is represented in the Q square. On the other hand, the heterozygous alleles like capital A and small a, it is represented in a way like 2pq, 2 into p into q. As we all know, in a population, in a species, there are homozygous individuals, there are recessive individuals, which means homozygous dominant, homozygous recessive, and some are heterozygous individuals. So these only create the gene pool. So to represent this, we have got a equation like p square plus q square plus 2pq is equal to 1. It is given by Hardy-Weinberg. This equation is very important. We'll solve a small problem also in further time. So there are also certain factors which affect the equilibrium, which means when these factors act, the Hardy-Weinberg equilibrium will not be applicable. When mutation will be there, if there is a mutation, the gene frequency may change, the allele frequency may change. So the particular frequency will not transfer from parents to their offspring. And recombination, which means recombination allows several genes to transfer, whereas linked genes are just transferred in a closed way. Then if there is gene migration, which means in a population, if the individuals are migrating and different individuals are coming in that particular population, so then also this Hardy-Weinberg equilibrium will not be applicable. And genotype, if there is any elimination of genes, loss of genes, then also this Hardy-Weinberg equilibrium should not be applicable. So when this Hardy-Weinberg equilibrium is not applicable, when mutation is there, recombination is there, gene migration and genetics. These are the important factors which basically affect the equilibrium. Now we'll solve a problem. Just look at the question. In a population, the number of individuals having genotype homozygous dominant, that is capital A is 0.7. On the other hand, the homozygous recessive is 0.3. Find out the frequency of heterozygous dominant, that is capital A small a. So from the question we have got The frequency of homozygous dominant, that is capital A, capital A, 0.7, small a homozygous recessive, that is 0.3, but the heterozygous dominant is not mentioned. So again we'll put this formula, and we have already given the data of homozygous dominant and homozygous recessive, but we have not given this particular heterozygous one. Okay, so what we'll do, we will put here into 2 PQ the given data of P and Q. So 2 is equal 2 into 0.7 plus 0.3. So it will come like heterozygous individuals are 0.42. Okay. Now we can recheck it. So now we have received the data of heterozygous dominant, that is 0.42, and how it came by multiplying this P and Q data into 2. Okay, understood? Here actually plus will be placed. Just wait a second. By mistake, I have mentioned here. So here the multiplication will be occur. So when you will multiplicate, it will come like 0.42, and we can recheck it by putting in the same formula, as we have given the data of homozygous individuals dominant and recessive one like 0.7 square, 0.7 will do it square, and here also 0.3 will do it square, and we have received the heterozygous individual data, that is 0.42, and we'll try to match with 1. So when you will calculate this, you will get the answer like here given data will be 1, and here also the given data will be 1. So now it is proven that, so this is the frequency of the heterozygous individuals. Thank you.